Well, thanks ever so much for the kind invitation to speak. So I'd just like to talk to you today a little bit about some work I've been doing on using mathematical models to elucidate the mechanisms underpinning a group of inherited retinal diseases, which are known collectively as retinitis pigmentosa. So just some brief acknowledgements. This work has been conducted in part with the people at the top, funded by the institutions in the middle, and conducted at the institutions at the bottom of the screen there. So I wanted to begin with a couple of definitions. So I work within a field for which I've coined the term mathematical ophthalmology. And I define this term as using mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease. So using mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease. And this is in contrast to an existing term called computational ophthalmology, which is about using computational algorithms to preserve vision by identifying and predicting ocular features and risks. So the various similarities and differences between these two fields, all three of them involve understanding prediction and treatment. So both of them involve trying to gain a deeper understanding than you would be possible through experimental or clinical techniques alone. They involve predictions, so making predictions as to how a healthy homeostasis is maintained, or to what will happen to a system under perturbations or in disease, and also about thinking about identifying novel drug targets or improving treatment strategies or personalizing treatment. But they do these in different balances, and each of them have their own strengths and weaknesses. In terms of the strength of mathematical ophthalmology, the strength there is that you get insight into the mechanisms that underpin, say, maintenance of a healthy homeostasis or what's going wrong in disease. By contrast, computational ophthalmology, by which I mean artificial intelligence type techniques, has either low or no mechanistic insight, because it doesn't contain within itself descriptions of the mechanisms in question. On the other hand, computational ophthalmology is an incredibly powerful predictor, in part precisely because it doesn't depend upon these mechanistic descriptions. In contrast, mathematical ophthalmology, by which I mean mathematical models, are rather weaker on prediction, in that they depend upon having accurately captured the underlying mechanisms in order to make an accurate prediction. So these two sort of fields are not, in competition with each other. Each of them have their strengths and weaknesses, and they complement each other quite nicely. So I work very much within mathematical ophthalmology, although I'd be keen to combine these two together more in the future, and I'm seeking to encourage others to do so also. And if you want to talk to them about them together, I've come up with the acronym MACO for mathematical and computational ophthalmology. So that's just a sort of background in terms of the field in which I work. A little bit of background biology, because I realise not everyone here will work in the eye. So the diagram you're seeing, I'll just see if hopefully you can see my pointer up, put a laser pointer up. So the diagram on the left-hand side here is of the eye viewed in the transverse plane, so cut through horizontally. At the back of the eye, the key layers are the sclera, which is the white rubbery outer coating. You've then got a layer called the choroid, which is a vascular layer which supplies the retina with oxygen and other nutrients. And then the retina itself, which is the light detecting layer, which runs from the ora serrata around to the optic disc. So the retina contains a number of different cell types, but in particular the photoreceptors, which are the light detecting cells. And those come in two varieties, rods and cones. Rods are for your low acuity monochromatic vision, peripheral vision, and cones are for high acuity vision, central vision, color vision, and cones are very much used in well-lit conditions, and rods in poorly lit conditions. So these cells are distributed pretty heterogeneously across the eye. So this is shown in the diagram on the right-hand side here. So the eccentricity, 0 degrees eccentricity, corresponds to the fovea, which is just opposite the lens. Then there's an increase in eccentricity that goes around to the ora serrata, either nasally or temporally. So you can see that cones are very densely packed in the fovea and then drop off pretty rapidly as you move towards the ora serrata, whereas rods are absent from the very center of the fovea, from the foveola, and then increase in density as you move towards about 20 degrees eccentricity and then drop off more gradually. So the fovea is cone dominated, and the rest of the retina is rod dominated. So in the work I'm going to talk to you about today, I'm looking at this group of diseases called retinitis pigmentosa. So these are inherited diseases, and the mutant genes that are involved are typically expressed in the rod photoreceptors, occasionally in the underlying RPE, retinal pigment epithelial cells, but typically not in the cone photoreceptors. If you're a patient suffering from this condition, then you'll usually experience night blindness and poor dark adaptation in adolescence, and then you'll begin to lose peripheral vision in your early adulthood, developing into tunnel vision and eventually loss of central vision by about the age of 60. This is a leading cause of blindness worldwide, so currently about 1 in 3.5 to 1 in 4,000 people are affected, so that's about 2 million people, and it's the most common inherited retinal disease. And it's currently incurable in that we can't completely halt or reverse the disease, although there are treatments available to limit the progression. One of the things that makes it difficult to develop effective treatments is that we don't really understand fully the mechanisms that underpin the disease. And one of the reasons it's difficult to get a handle on those mechanisms is it's hard to experimentally isolate them and to understand what each mechanism, what that causes in terms of the causal chain for pathogenesis. But while that is difficult experimentally, it plays very much to the strength of mathematical modelling, because we can use mathematical models to isolate mechanisms and to ask the question, well, if this was only a particular mechanism was at play, then what would the effects be? So there are two particular features that I want to highlight about RP, retinitis pigmentosa, that are relevant to what I'm going to discuss next. The first is that it's a rod cone dystrophy. So this means that the rod photoreceptors are affected earlier and more severely than the cones. So it's to be expected that the rods would be affected given that they express the mutant gene. However, as I said, the cones don't express this gene, and yet they degenerate have a secondary wave of degeneration that follows the rod degeneration. So the question then arises, why do the cones degenerate? They would seem to depend either directly or indirectly upon the rods for their survival. So a number of mechanisms have been hypothesized to explain secondary cone loss, but we don't know, and these are complementary mechanisms, but we don't know which of these are most important. Alongside this, although typically discussed separately in the literature, we have the fact that degeneration initiates in the mid or far periphery, usually in small patches, and these then expand and coalesce, giving rise to a number of very characteristic spatiotemporal patterns of retinal degeneration and hence visual field loss. And so my suggestion is that the mechanisms that people have put forward for the secondary cone loss would in fact also explain these patterns of visual field and retinal degeneration. And so we're going to be using that in the modelling that goes that follows. So I mentioned there's a number of different mechanisms that have been proposed to explain secondary cone loss and that could also explain these patterns of degeneration. So in short order, these are the oxygen toxicity mechanism, trophic factor depletion, metabolic disruption, toxic substance release, and microglia activation. I'm afraid I don't have time to go through these in detail, but these are, as I said, complementary mechanisms, so it could be any of these or any combination of them. And then on the other hand, we have these very characteristic patterns of retinal degeneration and hence visual field loss, which are represented on the right-hand side here. So the black regions here are blind spots or scotomas, and the white regions are regions of preserved vision. And so the categorization here comes from a paper by Grover back in the late 90s, where they used Goldman visual field tests and grouped on RP patients and grouped them into 3 groups and 6 subgroups based on the patterns of visual field loss, which I've represented in this sort of cartoonized type diagram. And so the question is, which of these patterns could be explained by which of these mechanisms? And in what follows, to keep things brief, I'm going to focus on the oxygen toxicity mechanism. And so the process is as follows. I'm going to isolate a particular mechanism with a mathematical model in a way that would be difficult to do experimentally, namely the oxygen toxicity mechanism. And then I'm going to ask the question, if only that mechanism was operating, what patterns of degeneration would result? And in so much as this mechanism is able to recapitulate the patterns you see on the right-hand side, and that shows the theoretical sufficiency of that mechanism, although it doesn't show it is the mechanism that's creating the patterns. And perhaps more interestingly, insofar as it's not able to reproduce those patterns, then it reveals the necessity of other mechanisms, perhaps the other ones that I've listed on the left. So focusing on the oxygen toxicity hypothesis then. So the way this works is you start with a mutation that is usually expressed by the rods, so leads to the degeneration of rod photoreceptors. When you lose rod photoreceptors, that decreases the rate of oxygen uptake, which results in an increase in oxygen levels. If these increase enough, then they become toxically high for neighboring photoreceptors, which leads to further photoreceptor degeneration. And then you get a sort of self-reinforcing cycle as further photoreceptor loss leads to a further decrease in uptake of oxygen and hence an increase in oxygen levels, which leads to further photoreceptor degeneration. So there's a number of lines of experimental and clinical evidence pointing towards this mechanism. So, for example, experiments using oxygen-sensitive microelectrodes in rats and cat models of RP have shown elevated oxygen levels. In humans, we see attenuation of the retinal capillaries, indicating increased oxygen levels. We know that the choroid, this is the vascular layer that supplies the outer retina in which the photoreceptors reside, that this autoregulates very poorly in response to increased oxygen levels, so you keep having a supply of oxygen even when oxygen levels have risen. It's been shown in pig models of RP that oxidative damage is done to lipids, proteins, and DNA. And on the flip side of that, we have a number of experiments showing the efficacy of antioxidant therapies in neutralizing the reactive oxygen species. So it seems that regardless of which other mechanisms are at play, oxygen toxicity certainly seems to be an important factor in this disease. And so what I'd like to do in this presentation is pose two fairly straightforward modeling questions. The first is, under what conditions will a patch of degeneration spread? So I said degeneration starts in small patches which expand and coalesce. So sometimes those patches will remain fixed in size, sometimes they expand. Can we explain that by this mechanism? And the second question is, which patterns of degeneration are explicable via this mechanism, referring back to the patterns I showed you earlier of visual field loss. So I'm going to formulate a mathematical model. I realize that most people here are not mathematicians, so I'm not going to go into all the detailed equations. I'm just going to explain it at a more kind of conceptual level. If you are a mathematician, this is a reaction diffusion partial differential equation model. But all you really need to understand in terms of what that means is that I'm going to be modeling the density of photoreceptors and the concentration of oxygen and the way that those evolve through space and over time. So I'm going to be doing that on a domain or a region of space which will span the back of the eye, going from the fovea at the angle theta equals 0 around to the ora serrata at theta equals 1, where I've normalized the angle here to go between 0 and 1. So these are in what's called spherical polar coordinates. Now it's important to incorporate into this model the distribution of photoreceptors. And so here I'm using some data from Christine Curcio's lab from 1990, where she measured the distribution of rods and cones in a healthy human retina. So in order to incorporate that into the model, I then need to fit some mathematical expressions to the data. So this is what these expressions are at the bottom. So the top one is an expression for the distribution of cones, and the bottom is an expression for a distribution of rods as a function of theta, this angle around the back of the eye. So the top left panel here is the rod density. The black dotted line is the data. The red line is my fit to that data. And so the x-axis here is the eccentricity going from the fovea to the ora serrata. So you can see there's a decent fit between rods and data and approximation. Similarly for the cones, and then if you add those two panels together, then you get the total photoreceptor distribution. So then in terms of developing the model, so that the main meat of a model consists of what are known as governing equations, and there are two of these for the model in its simplest form. The first equation deals with the rate of change of the oxygen concentration within the retina and how that evolves through space and over time. And so to account for that, we're looking at the diffusion of oxygen as it goes towards the ora serrata or towards the fovea, the uptake of oxygen by photoreceptors, and its exchange with the choroid, which supplies oxygen to the retina. And in the second equation, we'll look at the rate of change of the photoreceptor density and how that evolves through space and over time. And what we'll say is that the photoreceptors remain fixed in density and healthy unless and until the oxygen concentration comes to exceed what I call a hyperoxic threshold. So oxygen levels become toxically high. I call that threshold secret. So if that happens, then the photoreceptors will degenerate exponentially in that area. And then to close this system, we need two other pieces of information. Firstly, we need some initial conditions. And what I'm going to say is we're going to remove some photoreceptors from some interval theta between theta 1 and theta 2. So here I'm representing this in this diagram. So here, again, theta is going from the fovea to the ora serrata on the x-axis. Theta 1 and theta 2 are these two blue vertical lines. So I'm removing a patch of photoreceptors from between these two lines. And that's why the red line here, which is the photoreceptors, goes down to 0 in that interval. And then as a consequence, the oxygen level comes up to very high sort of choroidal levels in the interval in between. And then lastly, to close the system, we need things called boundary conditions, which says what happens at the edges of our domain. So what we're saying is that oxygen can't escape from the fovea because of the symmetry of the problem. And then for simplicity, we're saying it can't escape from the ora serrata. So then to summarize, the inputs to this model are the boundaries of this patch of degeneration and the hyperoxic threshold, and the outputs are the evolving concentration of oxygen and photoreceptor density through space and over time. So going back to the questions that I posed then, the first question is, when will a patch of degeneration spread? Well, to explore that, I'm going to go back to this diagram that I showed you a moment ago. where we've taken out a patch of photoreceptors. And so simply put, or relatively simply, a patch of degeneration is going to expand if and only if the oxygen concentration on the border of the patch of, or one of the two borders of the patch of degeneration exceeds the hyperoxic threshold. So imagine, for example, that happened on the right-hand boundary here, that the oxygen concentration on this boundary is above the hyperoxic threshold. That would mean that there's some interval to the right of the boundary, in what's currently a healthy region, where the oxygen concentration is also above that threshold. The consequence of that will be that it will cause the photoreceptors in that region to degenerate, which will cause then the expansion of this patch in the rightwards or peripheral direction. And similarly, you'd have the same thing at the left-hand boundary. If, on the other hand, the oxygen concentration is at or below the hyperoxic threshold on the boundary, that means that the oxygen concentration outside of the patch will also be below that threshold, and so the boundary will remain fixed. And that means then that you have four possible cases as to what can happen. Either it's the case that both boundaries are fixed in position, or you'll get expansion only towards the center of the retina, or only towards the periphery, or both centrally and peripherally. And so these cases are represented in this diagram on the right-hand side. Now, if you don't like this diagram, don't worry, you don't need to understand it to understand the talk, but I quite like this as a way of explaining the possible cases. So this is a representation of θ1, θ2 parameter space, so based on the position of the left-hand and right-hand boundaries. And you'll notice it's upper triangular, and this is because, of course, the left-hand boundary θ1 is always to the left of the right-hand boundary θ2. And so you can see these four cases that I described represented here. So the green regions here of the θ1, θ2 parameter space of type A are stable regions where the patch will remain fixed in position. The orange regions of type B are ones where it's going to expand centrally only. Regions of type C are where it will expand peripherally only. And then regions of type D are where it should expand both centrally and peripherally. Another perhaps more intuitive way of representing this would be to say that what this means is that we can actually divide up the retina into an alternating series of stable and unstable regions. So by an unstable region, I mean if the boundary of the patch of degeneration lies in that region, it will expand outwards until it reaches either a stable region or the edge of the retina. Whereas if a boundary of a patch is in a stable region, it will remain fixed in position. So comparing this to the photoreceptor distribution, what we have is a peripheral unstable region, a mid-peripheral stable region, a para or peripheral unstable region, a near-central stable region, and then if we zoom into the central region, there's also, as shown in this inset, a central unstable region. Or to look at it a bit more like an ophthalmologist might see it. So if we look at it, the retina en face, as if you're looking through a fundoscope, then you have this sort of bullseye pattern of alternating stable and unstable regions. Now, the results I've shown you here are actually for the model in what's called its steady state, so where things are not changing in time. So to confirm that the results that I've shown you are indeed accurate, we need to compare that to the full dynamic model, the model that changes in time. So I'm going to show you a few simulations, but just to motivate those, I'm going to show you a couple of examples so you kind of know what it is you're looking at. So if you look at this diagram on the right-hand side, this is a heat map, which shows the density of photoreceptors. If it's red, then that's a high density. If it's blue, that's a low density. On the x-axis, we have the eccentricity increasing from the fovea to the ora serrata, and then time is marching upwards. So here what I've done is removed a large swathe of photoreceptors, between large patch of photoreceptors, and then this patch is expanded outwards. So it's moved outwards to the right towards the periphery, ora serrata, and also inwards to the left centrally towards the fovea. In contrast, in this diagram on the left-hand side, I've removed a smaller patch somewhere in the mid-periphery, and it's remained fixed in size. So both of these boundaries are vertical and don't expand outwards in any way. So what I'm going to do is look at the four cases that I mentioned earlier and see if the dynamic model matches with what I showed in the steady state case. So first of all, if we pick a region of type A, then what we're expecting is that the patch will remain stationary. And I just note that a patch of loss actually corresponds to an annulus in 2D because we're looking at a 1D model. So as we expect, if we have a point from region A, then it remains fixed in its position. And on the left, so the right-hand side, I'm plotting photoreceptor density. The left-hand side is the corresponding oxygen concentration. So the oxygen concentration is high, where the photoreceptor density is low. If we move to a region of type B, then we expect that to expand inwards only. And again, that's what happens. So it goes inwards towards the fovea, but remains stationary on the right-hand side. If we have a region of type C, we expect it to expand outwards, but not inwards. And again, that's what we find. So stationary on the left, it expands outwards on the right. And then lastly, if we're in a region of type D, we expect the annulus to expand both inwards and outwards. And again, that's what we get. Before moving on to the next question, I just wanted to note that the speed of degeneration, the wave speed as the degeneration propagates outwards, is not constant. So if it was constant, this would be like a triangle in the corner here, for example, and you have a straight line, but actually it bends around to the edge, so the speed accelerates here. And similarly on the left, it sort of accelerates and then deaccelerates as it moves towards the fovea. So I haven't got time to talk about this in detail, but it turns out you can actually come up with a mathematical, this is the last equation you'll see, mathematical expression for the speed of propagation of degeneration, which is what I'm giving here on the right. It might look complicated, although from a point of view of a mathematician, this is a fairly simple equation. And the nice thing about it is that we know the values of the parameters on the right-hand side. So that the reason this would be quite useful, say, to a mathematician, to an experimentalist or clinician, is that if you want to solve the simulations I'm showing on the left, that takes hours of computational time. Whereas if you say put this formula into an Excel spreadsheet, you can solve this in a fraction of a second and quickly get an answer as to the rate of propagation. And this incidentally shows that the rate of propagation of degeneration is a monotone decreasing function of the local photoreceptor density. So if the photoreceptor density is lower, the wave of degeneration will propagate faster. So that answers the first question as to when a patch of degeneration will spread. And then I'll finish with the second question, which is which patterns of degeneration are explicable purely via the oxygen toxicity mechanism. So just to remind you, these are the patterns of degeneration that we see from measurements. So you might start with a restriction of the peripheral visual field, perhaps accompanied by a little ring scotoma around the fovea. Sometimes it will start in the superior field and wind around inferiorly. Sometimes it starts from the mid-peripheral ring scotoma, and that extends superiorly or inferiorly and winds around the edge. In each case, you'll end up with central vision preserved, but eventually that's lost. So now we're going to move from a 1D model to a 2D model. So as well as having a domain that goes from the fovea to the oris aorta in the polar direction, we're now also going to flip around the z-axis in what's known as the azimuthal direction, and so create a model which is posed on a 2D shell. So another way to represent that is on the right here. So this is the 2D shell, and I've painted onto it the photoreceptor density. So red here is high and blue is low. And the simulations I'm going to show you, I'm going to project this 2D shell down onto a sort of 2D shell in 3D space onto a 2D plane, and then flip that up on fast so you can see everything that's happening. So what I'm going to do now is just damage but not remove photoreceptors from a region and see what happens next. So here I've damaged some photoreceptors in a region of the mid-periphery at some time, and then later on this actually recovers. So under the oxygen toxicity mechanism, the retina is very robust to damage in the mid-periphery. By contrast, if I take out or damage a patch towards the periphery, then actually something rather different happens. So as time marches onwards, this then consolidates, it expands out towards the far periphery, and then begins to expand out in two arms around the edge of the retina until those arms meet on the far side. And so then this actually replicates the peripheral pattern that you see at the mid stage of pattern 3 that I described earlier. Another thing we might consider, rather than damaging the retina, if we start with simply a healthy retina, we might include alongside the oxygen toxicity driven loss of rods and cones, we might also include a mutation induced loss of rods, since the rods will die regardless of oxygen toxicity, simply because of the mutation that they're expressing. So what I'm plotting here then is the cone density under these conditions, taking the log of the cone density just to make visualization easier, and how that evolves over time. So what we find here is that we begin to lose cones around the periphery, and that expands inwards in a wave. And then later we get a new patch of degeneration that starts around the fovea and expands outwards, meeting the inward traveling wave somewhere in the mid-periphery, until all you've got left is a little red dot somewhere in the center representing the fovea. And so that replicates pattern 1B that I described earlier. So just to summarize that then, comparing the in vivo patterns we see in humans with the in silico patterns from my simulations, we've found that under the oxygen toxicity hypothesis, we can recapitulate pattern 1B. We can also recapitulate the last stage of pattern 3, where you have degeneration spreading around the periphery. But what we're not able to reproduce are patterns of type the early stage of pattern 3, or generally pattern 2, where there's a preferential loss of mid-peripheral photoreceptors. So to summarize them, the models that I've developed here are the first to consider the oxygen toxicity hypothesis for retinitis pigmentosa, or to account for the heterogeneous distribution of photoreceptors across the eye. We've been able to determine the retinal topographical regions in which a patch of photoreceptor loss will spread, and that's determined by whether they lie in one of these alternating regions of stable or unstable regions, that bullseye pattern I showed you earlier. We've been able to predict that the wave speed of degeneration is a decreasing function of the local photoreceptor density, and have a simple formula that you can use to quickly calculate that. And we've predicted spatiotemporal patterns of retinal degeneration, demonstrating both the strengths and limitations of the oxygen toxicity hypothesis. So on the one hand, the hypothesis is strong in so much as it's shown to be sufficient to recapitulate some of the patterns that we saw, but actually, I think more interestingly, it shows that there are some patterns of degeneration which are theoretically inaccessible to that mechanism, demonstrating the necessity of other mechanisms being at play there. And just to briefly tell you how this is in current and future work, how we're going to expand this. So I've shown you here today that the oxygen toxicity mechanism gives rise to these sorts of patterns of degeneration. I've also looked at the trophic factor mechanism, which gives rise to this sort of pattern. And I've got current and ongoing work looking at metabolic dysregulation and toxic substance microglia hypotheses, which suggests, which I'm predicting will give rise to the patterns of degeneration I've shown here. And so you can think about then, but not just modeling these independently, but also thinking about how they might combine together to give rise to the full suite of patterns of degeneration that you see in patients. So for instance, pattern 1 might be a combination of oxygen toxicity and trophic factor mechanisms. Pattern 2 could potentially involve all of the mechanisms that I've described. And pattern 3, again, could involve a subset of those mechanisms. I'm not saying that these are the only ways that this could occur, but that the important point that I'm trying to make here is that this has important implications in terms of treatment if you're a clinician, because what the modeling suggests is that different mechanisms are responsible for the degeneration of the retina at different temporal points in the disease and at different spatial locations. So if you're thinking about treating the condition and you want to prevent further photoreceptor loss, then you're going to have to take into account which mechanisms are driving the disease at different temporal phases and in different spatial locations. And before I finish, I just wanted to tell you if you're a couple of opportunities that some of you might be interested in. So I'm currently guest editing a special issue in a journal called IOVS, or Investigative Ophthalmology and Visual Sciences. This is an issue in mathematical and computational ophthalmology. So if you're working in those fields and have a paper that might be appropriate, then do consider this special issue of publication. It's published in the normal way in the journal, but just gets extra impact by being included also in the special issue. And the second thing is, if you'd like to learn more about mathematical and computational, I should mention that there's a number of invited reviews from various luminaries in the field plus me. And then if you'd like to learn more about mathematical and computational ophthalmology, then I've got an online seminar series, and the talks go out live on YouTube, but the recordings are maintained as well. So there's a number of past talks from people on the mathematical side on the left here and computational side on the right. And the web link for that's just there. And if you the next talk is going to be given by Giovanna Guidaboni on Tuesday, the 14th of November at 2 p.m. GMT, and she'll be talking about mathematical and computational modeling of ocular hemodynamics. So I'll just finish on this slide just to say I've been talking to you about some of my work today on inherited retinal degenerations. There's other ongoing work there that I have worked in the past and continue to work on oxygen distribution and retinal metabolism, AMD, but in its treatment and disease pathogenesis, and also visual neuroscience. And this is sort of my eye work. Outside of that, I work on various things in biomedicine, including microbiology and oncology. So if you'd like to learn more, then do just check out my webpage, and I've got a Twitter handle there. So thanks ever so much for listening, and I'd be happy to take any questions that people might have.